Majority of it after the on Friday. Yeah.
So we're going to be starting this section on metabolism, and your notes are really detailed on this.
Yeah,
what I'm going to try and do is simplify it into more of key points on here with it. But you have the details in here, so if you need to go back and refer to it, you've got a lot of detail to work with in there, as well as your book. This is not something where, I know that sometimes it doesn't work real convenient to study something every day in your life. Inconvenience yourself on this one, because metabolism is one of those things that you may have to really take this small bites at a time to have it make sense, and it's hard to remember it if it doesn't make sense to you. So I'm just letting you know now that if you procrastinate on this, it will be ugly. There isn't anybody who gets away with procrastinating on this, and it's okay. Did they get it fixed? Why don't they have you in there then? So you'll have to contact distance learning afterwards, because that's where it's showing you dropped.
Maybe that one person made the mistake of hitting the button.
So on here, if you have questions as we go, let me know. You'll probably have to really speak up today, because my voice is not going to do well for talking really loud. I suspect by the end of the day I won't be talking anymore at this rate, but I'm also not hearing very well, and when I hear things, it seems to get warbled into you saying something inappropriate. So if I think that you said the wrong thing and I didn't get the communication wasn't clear, just correct me on there. Yesterday I was really kind of like, wow, I can't believe they said that, and I'm like, they just are like, you heard wrong.
Your ears are clogged from being sick.
It's not pretty right now. So with cellular respiration, we've got fermentation and aerobic pathways that are going to be part of this. The fermentation involves partially degrading the sugar in there, and it occurs without the use of oxygen. So it's not broken down all the way. It's just broken down part way. With aerobic respiration, oxygen is going to be involved in it, and it's broken down all the way to make carbon dioxide at the end. So you have nothing but carbon dioxide and water left at the end of the process. Cellular respiration is a term that technically includes both, and this came about because of the relationship to breathing oxygen with doing metabolism in there. And I don't know why they still even use the term respiration in there. It's just a lot easier to refer to it as aerobic metabolism, but I put terms in here to try and stay consistent with your book in there so that it made more sense to you. But you will see metabolism replaced instead of respiration in other books out there. So basically the overview of what we get with metabolism is you start with some sort of organic compound, mix it with oxygen, breaks down to carbon dioxide, water, and energy. So what we look at is the main pathway in animal cells is glucose. Yes, you consume things other than glucose, but they will join in where glucose is in the pathway. So you have your glucose plus oxygen, there's carbon dioxide, water, and energy in the form of ATP. So it is a spontaneous reaction. We've got a negative delta G of 686 in there, but you are still going to have to get it over that activation energy. So even though it's spontaneous, it isn't that it's just going to break down on its own. That keeps it under control with that activation energy. Your catabolic pathways are not going to directly perform cellular work. So instead, it will give you ATP that acts like a chemical drive shaft or an energy currency in there so that you can use the ATP to fuel things. So it's kind of like saying when you go to work, work doesn't turn around and give you your groceries and a place to live. They give you money, so you go buy your own groceries and a place to live with it in there. And that's what the ATP is going to be like. So there are all going to be a series of have what we call redox reactions. So redox is the term that I've seen used most frequently. That's what they refer to them in chemistry as. They're oxidation and reduction. So you'll see oxidation, reduction, reduction, oxidation. Any combination of there as ways of naming these reactions. And what they involve is transferring electrons in there. So oxidation is going to be the loss of electrons from one substance to another. Reduction is the gain of electrons in there. The way I remember it, being that you're going to reduce and gain, doesn't kind of click, is that it's going to reduce the overall negative number in there. Electrons have a negative charge. If you put more electrons on there, it becomes more negative. The overall value is reduced because it's more negative. That's how I remember it in my head. There. So the reducing agent is going to be the one that is the electron donor. Oxidizing agent is the electron acceptor. If you're going to move electrons from one substance to another, you're going to have both reactions occurring at the same time. You can't gain electrons somewhere out of nowhere. Something else has to lose electrons for that one to be able to gain it. So they occur together. And this diagram here shows that. So you have, in this particular case, the methane becomes oxidized to make carbon dioxide in here. The oxygen becomes reduced to make water. And you can't have this part occur without this, because otherwise there's no oxygen and no electrons in there. They just have to go together in there to make that. And so your electrons are going to lose some energy along the way as they're moved in there. And that's how you're going to get energy out of the reaction. So in metabolism, we've got this substance NAD in there that's going to go between an oxidized and a reduced state in here. And so rather than this reaction of reducing water or oxygen to make water to be a really explosive reaction in terms of energy in there. So rather than having it be really explosive, if you look at it and you go, okay, the water can go from point A to point B down a waterfall, or it can run around a little pathway and come down, and it's going to be a lot more controlled coming down. By using the NAD, you can harvest the energy in a lot more controlled pattern by passing along some NADs in there.
What's the difference between the NAD and what was the other, the A?
NADH?
Yeah.
So one is the oxidized state, one's the reduced state.
Okay.
So we'll alternate between the two of them. So NAD is nicotinamide adenine dinucleotide. This name in here, you'll probably never need to use for anything. We just always refer to it as NAD in there. So an enzyme is going to basically remove the hydrogens from it to oxidize it in there, is what will happen. So this is the most common and most versatile electron acceptor in respiration in there. So you're going to see NAD a lot in these reactions in there. So as you start to pass things down the hill, what you're going to see is that each neighbor is slightly more electronegative than the uphill neighbor to help move the electrons down the hill. So you have to go from the higher energy state to the lower energy state. It's going to roll down the hill. Ball doesn't roll up the hill. It's going to roll down in there. So when we look at the reactions that we have here, this picture is a good one to come back to. That gives the overall big picture of going, okay, what order are these things happening in? You've got your cell here, and the first step is going to be glycolysis in there. With glycolysis, what you're going to be doing in the cytosol is you take glucose, you're going to split it in half to make two pyruvates. When you do that, you're going to get a little bit of energy out of it, is ATP. You're going to have some electrons get passed down to the end of the line and be used down there. This is first little bit of ATP that gets contributed to your bank. The next part has to go into the mitochondria to occur for the aerobic respiration. So you'll take the pyruvate, and it's going to get oxidized a little bit to make acetyl-CoA. And that will enter into the citric acid cycle. This will give you a little bit of ATP. It's going to give you a lot of electrons, but it's going to move on NADH. And then we do have another electron acceptor in there that's going to transfer the NADH.
Now, in the past, are we just going to need to know the definition on that, or just basically how it works?
The definition isn't going to want to go in your head till you know how it works.
Maybe draw it out.
Yes. So now you've got all these NADHs and FADH2s, and if you think of it as like you go to the grocery store with Canadian dollars and want to buy food in the United States, what's going to happen? They're going to look at you and say, this isn't our money, so change that. So if you tried to fuel your cells, your muscles using NADH, they'd be like, nice try, go get some ATP. So the electron transport chain is going to be where you convert the energy from this NADH into ATP at the end.
Are you recording this over there?
Yes. So you're going to get the barking cough that's going to come in from the
Oh, well.
So on here you'll notice some of these say substrate level phosphorylation and some of them say oxidative phosphorylation. This here, when we say substrate level, means that you're going to get the phosphate that goes on the ATP from another organic molecule. With oxidative, it's part of the redox reaction and it's going to be inorganic phosphate. It's just, there is a difference in where it comes from on there. It's still making ATP and you get the same thing out of it in the long run. So with your electron transport chain, basically what you're taking is this that could be the explosive reaction, and you're slowing it down through a series of steps so that you can harness that energy in a more controlled fashion, but you still end up with the same thing at the end. So out of the whole thing, what you're going to end up with is 38 ATP total, a net of 36 ATP. This net 36 number is the one that is going to come up the most frequently that you need to know, because you do have to put a little energy into it to get it started. So if you're thinking of it as you've got this ball that you need to roll down the hill, but you've got a little bump in the hill, you've got to put a little ATP in to get it over the hill.
Just like activation energy?
Yep. So that's why there is that difference of 2 ATP in there.
Activation.
It's your activation energy. It's the energy you have to put in to get it started. Kind of like when you go to make a barbecue, you've got to put in some energy, a little bit of lighter fluid, a little bit of fire from the matches, and then you get a whole lot of energy out of this cookie food. So when we look at glycolysis, it's broken down into 2 main phases in here. At the start, you have the energy investment phase. This is where you're lighting your barbecue here. You're going to have to put some ATP into it to get it started. Then you'll have the energy payoff phase in here, where you're going to get some ATP out of it. So you get 4 out of it. You have a net gain of 2, because you have to think of this was your investment out. If you invest $10 and you get $20 back, you didn't just make $20 and gain $20, you only gained 10. You still have to account for what you have to put into it. And then you're going to get 2 NADH out of it, and it's split in half to make 2 pyruvate and 2 water. So just down at the bottom here, it gives you your kind of summary in here. Glucose gives you 2 pyruvate and 2 waters. You're going to get 2 ATP out of it, 2 NADHs out of it. So we're going to look at glycolysis in more detail. what you want to do is use the details so that this makes sense to you. Right now you're just looking at it and you're like, okay, that's nice. But we're going to go through the details. You don't have to memorize the details unless that's what it takes for this to make sense to you. If that's the way your brain works, I'm sorry, I can't change that. That's the way I had to work it. But this is kind of the level of detail that you would want to know is what's on this. But you're going to get more so that this will make sense. So here is the way this is in your notes. There's the little blue numbers here. And these words are explaining what's happening in detail in there. It tells you the enzyme that's used in there. You're not going to need to know the enzyme, but I thought as long as I'm making this, I want you to have it as a good reference for later on if you need it. So if you sell the book back, this is still good enough to refer back to. So here we've got this energy investment phase here. And when you look at it, you can see you've got 6 carbons. Each corner represents a carbon on here. So 6 carbons are in here. You start out with your glucose. You're going to take and put some ATP into it. So what that means is you're going to take one of the phosphates off of the adenosine triphosphate that becomes diphosphate, and you're going to stick it on the glucose here. Right here is where it is, the little yellow ball in there. And so that's why it's called glucose 6-phosphate, is because it's on carbon number 6. So now we're going to rearrange that a little bit in here. And it's still the same number of carbons in here. Still the same phosphate on there. It's just rearranged. So we have fructose 6-phosphate. So sometimes when you go to light the barbecue, you put the matches in it and you come back and you're like, oh, it wasn't enough. Same thing here. It wasn't quite enough. You got to put some more energy into it. Get another ATP. So now you're going to have 2 phosphates on there. And so this one is fructose 1,6-bisphosphate, meaning it's on carbon 1 and carbon 6. This means 2. So now we have enough energy in here that it's burning up and it's going to split in half. So how you notice sometimes when you make a fire in there and you've got the logs burning and then a log cracks in half? You still have the same amount of log, it's just cracked into pieces. So now they have cracked in half. You've got 3 carbons over here, 3 carbons over here. These can go back and forth between the 2 forms. And that's a reversible reaction. But this is the one that you use up. So when you do that with equilibrium, what that means is this one's going to get converted into this. So you can think of it as one takes this pathway and one does this. In there. It's never going to really go this way. That never really happens in a cell, even though it technically could. So what you end up with is the glyceraldehyde 3-phosphate. You've got 3 carbons, but you've got 2 of those, so all your carbons are still accounted for at this point. And the phosphate on there. So it kind of makes sense when you look at that why we call it an energy investment phase. You got to stick some ATP into it to get your fire going and split it in half. So the splitting reactions, that's what they refer to as these, where it splits in half and then goes between those. So now we have the energy payoff phase in here, where we're going to take that glyceraldehyde 3-phosphate in here, and we're going to rearrange things a little bit to make this right here. And we're going to end up making 1,3-bisphosphoglycerate. So you're going to move some electrons here to this NADH. So now it's your fire's going and you're going to get some heat out of it in here. So you're going to get some ATP here. So you're going to pull off, you have 2 phosphates here. You're going to pull off one of them and put it on ATP. Now you're down to 1. The reason this says 2 is because you have 2 of everything in here. because you split it in half. So you had the one glucose that came down, made 2 glyceraldehyde 3-phosphate.
Right.
So you have actually 2 sets of these going on next to each other, and that's why you get 2 H. So the 3-phosphoglycerate gets rearranged, the 2-phosphoglycerate gets rearranged to phosphoenol pyruvate, and then you're going to pull off this last phosphate here to make 2 more ATP because you've got 2 sets of those reactions, and you end up with 2 pyruvates at the end. The pyruvate has 3 carbons, you've got 2 of them, that makes a total of 6 carbons. So all the carbons are still accounted for in here. So this one is anaerobic, it doesn't involve oxygen. It will occur whether or not oxygen is present in there. So does that make a little bit more sense what we mean by energy payoff in there? You're pulling your yeah, you got your fire going, and now you're releasing that heat off as ATP
And it
in there.
basically just divides, well, it splits it, whatever goes on top of, well, you split it, basically. Is that how it?
Yeah, it's like you start with
2 on top, and you definitely have 2 on the bottom, or what? Well, 2.
So you start with glucose up here.
Okay.
Here you've got the glyceraldehyde 3-phosphate. You end up with 2 pyruvate
Okay, but how many glucose do you
at the end.
I got that picture.
So when it talks about showing that you get 2 ATP off, it's because you've got 2 of these going on next to each other.
Okay.
So when you're looking at it, you go, well, wait a minute, I only see 1 phosphate coming off. How are you making 2 ATP? It's because you've got another one
Okay.
here, and the sum is 2 of them that comes out. There is some videos that are on Canvas on there. In that section where it has like videos and additional pictures, they're in there.
You're not the only one, so don't worry.
Yeah, this is one of those things that I was completely serious in saying that you're going to have to look at this small bits at a time.
But the test is next week.
You've got over a week.
So wait, so what chapters does that take on there to what?
12.
That's not bad. I'm seeing that. I'm going to be absolutely here, okay?
Marry me.
What I would do is you want to be able to, you've got the words on here. This is a Word document. You can pull off these pictures and put them on a separate paper and look at them in there if you wanted to. Or draw them, that's helpful too, because the more you write it, the more it makes sense.
Do we just,
And if you can look at the picture and find somebody who is a willing participant, probably from class, this will scare away your significant other really fast in there. And if you can sit down and explain it to them. The other option is if nobody wants to listen to you and you have no friends that owe you anything, talk to yourself. Those stuffed animals you had when you were a kid, line them up and teach it to them in
I
there.
have no one.
Because if you could explain it to something else in there, if you have pets, you know, if you listen, dog, you're going to be the smartest dog around. You need to understand them.
Before that, do we just need to know,
It
like,
will help you to be able to explain it like that. If you're just sitting there saying it in your head, that's not the same. It uses more of your brain to produce the sound and say it.
but,
No one's looking in your house. Talk to yourself.
do we just need to know, like, okay, what it starts with? It's just like, okay, there's 288, well, 2 NAD or whatever, and then it
Yeah, so
breaks
these
it
summary
down.
pictures in here,
Just the
like
numbers
this,
and the words.
these are the ones you want to really be able to explain. Those last ones that I showed you that had all the details, that is just to
So we know.
make you accept it up here.
Okay.
But the picture before that, so if you could explain this one here, you want to be able to explain that to
Gotcha.
yourself.
Okay.
So do we have to be able to identify the substance that we feel like that was
It's similar format to what you've had. So this is another picture that you would want to be able to explain in here. So here's where you're going to go from your pyruvate that's in your cytosol to things happening in the mitochondria. This pyruvate here, your body can't store this pyruvate. So we're looking at one of the choices that pyruvate has for continuing to break it down. So what you can do is it's going to go get transported to a transport protein into the mitochondria. And when it does that, you're going to pull off one of the carbons. So you've gone from 3 carbons down to 2 carbons here. And you've added on coenzyme A. So now you have what we call acetyl-CoA. In the process of pulling off a carbon, you're breaking something down. You're going to release some energy in there, moving some electrons, so you're going to get some NADH out of it. Then carbon dioxide, so what would you suspect is going to happen to that? You're going to exhale it. So when you are exercising and doing more metabolism, you need to breathe more because you're making more carbon dioxide that you need to exhale. And this is one of the ways that you get that carbon dioxide. Why does it say that the microphone is not working? Well, it's supposed to be recording.
Usually there's a little number at the corner when you're recording. Number. There's usually there's a little
Yeah,
number right
it's
corner.
plugged in. Oh, I'm not wearing it. But it definitely is loud enough without that. Oh, now I'm not going to be able to walk over to the board either. That's going to suck. It's recorded from other classes on there. I mean, and if you just go to the YouTube that's on there, there's like 150 YouTube videos that's on my YouTube account that you can access. So you can see it a million different ways on there. Okay, so now that we have this acetyl-CoA, it's going to go into this cycle. So this is another one that I'm going to give you lots of details in here so that this cycle will make sense to you. Can somebody grab me a stick from back there? No. Thank you. Okay. I can't hit you either because I'm on a short leash. So we have acetyl-CoA here that it's going to come into this cycle that keeps refeeding itself. So you've got 2 carbons here. What it's going to combine with is this oxaloacetate from the last turn of the cycle that has from the last cycle in here. But it's an eternal cycle. You were born with this cycle and you're going to die with this cycle. So we're not worried about where the original oxaloacetate came from. Magic put it there. So this acetyl-CoA, these 2 carbons are going to join with these 4 carbons to make this citrate that has 6 carbons in there. So all the carbons are still accounted for in there. We rearranged this a little bit to make isocitrate. Still has 6 carbons. And then we're going to make alpha-ketoglutarate from it. Alpha-ketoglutarate only has 5 carbons. We pulled off one of them here. More carbon dioxide to exhale. And then we're going to rearrange it a little bit again to make succinyl-CoA. Succinyl-CoA only has 4 carbons. Pulled off another one here to exhale. When you pull those off, you're breaking a bond, you're getting some more energy, which is where this NADH comes from on both of those. What you're doing is you add 2 carbons onto it, you pull off 1 here and 1 here.
So now we're back to a 4-carbon thing that we just have to rearrange to get to here again, so that you can add another one back on. It's kind of like with a water wheel where you just keep adding water to it and it spins around.
But why do we pull off carbons there?
So that you're breaking them off, because when you split off a carbon, that releases a lot of energy.
Okay.
So the more carbons you put into this, the more energy you get. So we've got succinyl-CoA. We're going to rearrange this to be succinate. There's a lot of energy that's released in rearranging this one again, so it makes this GTP that we can then really easily convert to ATP here. So you get a little bit more ATP out of this again here. Succinate's going to get rearranged to fumarate. We're going to release some electrons in there. We're going to stick them on FADH2 this time. Fumarate gets rearranged to malate, malate back to oxaloacetate, releases some more oxygens in there. FADH is another carrier like NADH. The FADH2 is just another one in there. So if you think of like your NADH is kind of like having Canadian dollars, now you just got some euros too. You're going to have to exchange them in the end. So I know that you look at all these names, and without the background in chemistry, the names don't mean anything to you. It's going to make more sense to you if you look and go, I got a 4-carbon thing, a 2-carbon thing makes a 6-carbon thing. Broke it down to have a 5-carbon thing and released one of them. Broke it down to have a 4-carbon thing, released one of them. And now I got my 4-carbon thing again here. Not this one, but there will be more of an abbreviated one. But this is, with this picture, what you want to be able to see is how you lose those last 2 carbons from the acetyl-CoA. This allows you to burn them off. So rather than trying to look at names of all of those intermediates, look at it and see this is how we get rid of the carbon. Because remember, what you're doing with this is you're taking your 6-carbon sugar and breaking it down to carbon dioxide. These are the steps how you're getting the carbon dioxide off. So if you go back to this picture up here, that picture that we just went through in the detail explains why you get the NADH and FADH2 off of there. The only thing I don't like about this picture is it doesn't show where you're getting all these carbon dioxides in breaking it down, because I think that's kind of the missing link in having it all click together. So now you have all of these NADHs and FADH2s that you can't spend. No. Now we're moving on to the electron transport chain. The citric acid cycle is that one where you break off the carbons, the round one. Oh, hot. Yeah. So the electron transport chain is a chain of proteins that are going to pass electrons down the chain. That's how it gets its name. So you've got all of the little proteins have names, and they have little names of all the things in here. And we name the multi-protein complexes in there. I'm not worried about you knowing those names. What is kind of interesting to note in there is you've got iron sulfide in here. If you are deficient in iron, what's going to happen to you? You're going to be tired because you can't pass your electrons to make your energy from it. So that's kind of interesting to see in there. Some of these other ones in here, you're going to have B vitamins that help make up some of these complexes in there. You're going to have B complexes that make up and add to these. So those vitamins and minerals that you get from eating your fruits and vegetables, the reason that you need to have those things is they fill in the pieces of this so that you can transport your electrons and get the energy from it. If you eat food that's all dead from it, it doesn't really like to go through this cycle, and it just takes the shortcut to fat. Yeah, if you can't go through these cycles and you still consume the calories, you've got to put it somewhere. And your storage pockets are fat. So after you move the electrons all the way down this chain here, down at the end, the last protein that you've got in that membrane is this ATPase here. And they've actually worked out the detail of the structure of this protein in there, where you've got this little piece that's a rotor and one that's a stator in there. So your hydrogen ions come in and they get dropped into a little space on the rotor that spins it all the way around, and when it gets almost to the other side, it drops through another little space and gets put out the other side. And in the process of spinning this here, it's going to generate some energy that's going to take this phosphate and glue it onto the ADP to make ATP.
So are we just going to need to know what comes out of it?
Yeah.
And then what comes out of it?
Yeah, this is in the long run in there. So basically, the more electrons you have from NADH and FADH2, the more spinning of this thing that you do to get more energy out at the end. And they have looked at this and they're like, well, now that we have this little thing in the cell, this has been looked at as a possible way to generate energy, because this is the way some other mechanical things that we've built actually work, having this thing that spins around for generating energy. So they're like, oh, hey, maybe we could use cells to create an energy source for us outside of the body to power the world with. So those other ones were more detailed pictures. This one here, I think, is probably the most useful of the pictures in there. So it's showing that NADH, it starts passing its electrons early in here. So every time it passes it through one of these big complexes, the complex moves a hydrogen ion to the other side. So you get a positive over here, some negatives still down here. When you have positive and negatives that are separated, it's potential energy to get them to come together. So you're going to get 3 ATP out of every NADH because it passes it through 3 of these pumps. FADH2 cheats a little bit. 3, you'll end up, the exchange rate ends up being 3 ATP for every NADH. FADH2, you're only going to get 2 because it cuts in the line. Only is going to pass through 2 in here. So you guys have probably heard of coenzyme Q10, a nutrient. Right there. That's where it goes. So that's the kind of cool thing about these metabolic cycles, is all of these nutrients that you hear about taking, you can actually, when you look at it in more detail, see where they fit in the cycle. It's a lot of times headaches are from not having adequate oxygen and blood flow in a place. So when you restore the energy in there to it, it's getting its energy and its nutrients again. So this is showing it being moved along here. Over here is where you have that ATPase, that one with the rotor and stator, that it's going to bring it back down through there as it comes through. It does this. Also, your electrons come together here. Your final electron acceptor is the oxygen in here, so it comes together with some hydrogens and makes water along the way. So your summary of what that does in here, it gives you these numbers in here saying that, you know, the NADH is going to give between 2.5 and 3.3 ATP. We round it to 3. We round this one to 2. Those are just the numbers that get used for everything else. When we're saying 36 ATP, it's approximate.
Are you gonna mix? You're not gonna mix the numbers up or anything like that during calculating? You're just base it off of the grid?
Mix what numbers up? Say this one's 3, this one's 2. You get 36 ATP out of the deal. That one set of numbers is enough to use. Even in biochemistry classes, we don't usually beat it down this much in there to say, okay, you can potentially have between this much and this much in there. Because in the body, if you need one of the intermediates in there to do something else with it, you take it. So you get a little bit less out of that glucose. It's just, nothing is as straightforward because it's a dynamic system that's always changing. So hopefully when you look at this one then, this is another one that's a good one for kind of summarizing things. Yeah, the summary picture in there. Yeah, like I said, use these summary pictures as what you want to know. The detailed pictures in there are to make your brain accept this in there. So this has got it summarized in here, where with glycolysis you get the 2-pyruvate, 2-NADH, 2-ATP. You have to switch and make pyruvate into acetyl-CoA. It goes into the citric acid cycle. 2-ATP directly, 6-NADH, 2-FADH2. You've got the 2 from here. Goes through the electron transport chain in there. And you have your total that comes out at the end. So if you wanted to go through and add up the delta G's of everything, which I can't see why you would really want to do that, but you could in there. So to completely break down glucose, we know that the delta G of that is negative 686 in there. For phosphorylating the ADP to ATP, so the energy that you're getting from breaking down the glucose to make the ATP, you've got 7.3. And there for each of those, you're going to have 38 ATP in there. And so when you look at that ratio in there, you've got about 40% of your potential energy in glucose that ends up being transferred to ATP. So that's under standard conditions in there, that the reaction is about 40% efficient. In the cell, it's actually more efficient than that because the cellular conditions are more ideal for that to happen in. It's more favorable conditions for that. So your body is actually more efficient than that. We can't mimic that in the lab, though, because we can't create those exact conditions that are in your cell. So as you do metabolism, though, so when you exercise, you're doing metabolism, you get warm, you generate some heat to raise the body temperature. Right now, you can go in various different temperatures and be okay because this is what helps keep you warm, too. But when you do a lot of it, you're going to generate extra heat in there that you have to dissipate as sweat. So to give you an idea, the most efficient car is only about 25%. You're over 40% in there. So it's a really efficient system in there. So the other option, if you don't have oxygen available, is fermentation. And what fermentation does is you don't have oxygen and you don't have an electron transport chain. So you are going to have to move some electrons, so you're going to need some of the NAD in there. But you're going to break things down part way. And what you'll break them down part way to make is either alcohol or lactic acid. And you're not actually going to break them down to make alcohol. So some of the yeasts will do that in there. you're going to make lactic acid. Animal cells will make lactic acid in there. So it's not broken down all the way to carbon dioxide here. So basically you take the pyruvate, and one choice is you convert it to ethanol. You get a little bit of energy out of that, not a lot. And the NAD becomes, or the NADH becomes NAD to keep refeeding that cycle in there so you can keep doing it. Or you make lactate or lactic acid. And that's what happens with fermentation in there. So this picture shows basically you take the glucose, you get it as far as pyruvate. Here it's got a choice. If oxygen is not present, you go this way. If oxygen is present, you're going to go this way. It would rather go this way anytime it can because it's going to get a lot more energy out of it. It wants to use the most efficient pathway. So your cells are only going to do this if oxygen is not present. If you have some oxygen present, it will use the oxygen that is present for as much of it as it can, and anything extra goes over here. So when we look at the ability to use the oxygen in there, it varies. Some organisms are obligate anaerobes. They find oxygen toxic. In there, they'll die in the presence of oxygen, so they have to do fermentation in there. And there's bacteria that fall into that category in there. For humans, where you would run into those bacteria is when you have wounds that aren't getting a blood supply in there, they're not getting oxygen, and they can get anaerobic infections in there. If you've been around anaerobic infections, they stink. When you have fermentation going on, it's going to produce some gas as a byproduct. So sometimes they'll get little bubbles in there. If you have a human being that you give them lactose sugar that they can't break down, their gut tries to ferment it. They produce gas. It is too late at that point for the gas to come up this way. It comes out the other end. So a lot of times in nursing homes, they give elderly people milk. 70 to 80% of adults over 40 are lactose intolerant. So their nutrition department has basically set it up for you to breathe flatulence from old people. So the obligate aerobes, these can only do aerobic respiration. They can't do any fermentation. This is your brain cells. So if your brain doesn't have sugar, it dies off. Yeah. And they do. It's a whole thing with politics and milk in the dairy industry in there. So you don't actually really gain any calcium out of it, but you have people that make these decisions that have taken 1 5-credit, maybe, nutrition class that's based on USDA stuff. That's not enough to make decisions. There's a whole lot more to nutrition than what you learn in 1 nutrition class, but that's what it gets based off of. Yeah, you're going to hate it because if you, yeah. So facultative anaerobes, an example of these types of cells is in your muscles. They'll use aerobic respiration if they can, but if they can't, then they'll do fermentation in there. So evolutionary significance of glycolysis, they figured that the ancient prokaryotes probably used glycolysis to make ATP before there was oxygen in the atmosphere. Once you had the cyanobacteria in there, your early plants put oxygen in as a byproduct of photosynthesis. Then once you had the oxygen, there was the opportunity to do metabolism using oxygen. But if there's no oxygen in there, You can't do it in there. So in the prokaryote world, these are just a couple of the possible options for doing metabolism. So I had to take a 5-credit class that the whole thing we did was metabolic cycles like that of bacteria. Different metabolic cycles. Basically, any substance that is on Earth, there is a bacteria that has a metabolic cycle that can somehow break it down or make it in there. There is a lot more metabolic cycles than just that. So the versatility of catabolism in there. So while you're doing glycolysis, we looked at what happens with glucose in there, but other carbohydrates will get converted into glucose or their intermediates in there. And then they just join into the cycle. Your proteins, what you're going to have to do is you're going to break those down into amino acids. You'll use those amino acids to make new proteins. And then if there's some left over, some of them can be converted into the intermediates of glycolysis or the citric acid cycle. All of the nitrogen waste from any of those left over has to be processed through the kidneys, the liver and kidneys in there, makes ammonia and urea. So if somebody's prone to kidney stones and they have excess protein coming in, that's a lot more solids that the kidneys have to deal with, and it can trigger even more kidney stones for them. And then if you still have more than that, your body does not like to use amino acids and proteins to make energy. It will stop using everything else, particularly the fat, until it's dealt with those excess amino acids because of that nitrogen waste. It doesn't want that sitting around in there. And it'll just turn everything else to fat until you've gotten those used up. So the idea that you can eat as much protein as you want and it's not going to turn to fat, wrong. There's nothing you can eat as much as you want of. It can all turn to fat. So this kind of shows it here where you've got the carbohydrates. So they'll either come in at the top of glucose or somewhere in the middle. Proteins get broken down. You've got the nitrogen waste you have to deal with in another way. Some of them can come in here, some of them can come in here, some of them can come in here. Your fatty acids in here, glycerol is a 3-carbon sugar that comes in at glyceraldehyde 3-phosphate. But your fatty acids, all of those have 16 to 24 carbons. For a single triglyceride, you've got 3 fatty acids on there. So if you're breaking them off 2 carbons at a time, let's just say you've got one that's 16. 2 carbons at a time, how many acetyl-CoA are you going to get? 8. 16 divided by 2. So that is going to be 8 turns of this cycle you have to do for that fatty acid instead of 2. That's why fats have so many more calories in them. They have to be broken down in here. This is why you have to do aerobic metabolism to burn fat. It comes in right here. This stuff isn't going to do anything with the fatty acids. So one triglyceride has 3 of those 16 to 24 carbon fatty acids on there. So even being conservative, you're going to get 24 acetyl-CoA out of one triglyceride, at least, maybe more. 24 turns of the cycle. And what isn't on here is that this fatty acids pathway is reversible. So if you put in too much of anything, it can just make this. So for biosynthesis or making things in here, so you use the amino acids that you get to make new proteins. Some of the intermediates from the citric acid cycle and glycolysis get sent somewhere else. Excess food in here, it's all converted to fat stores in here. So if somebody tells you that there is something you can eat that's not converted to fat stores, it's probably going to make you have diarrhea and just run right through you and take some of your other nutrients with you, because biochemically, this is what happens. But most people don't understand that. And if you promise somebody, take this and you'll be skinny, they really want to be skinny a lot of times, and they'll pay anything for it. then you disappear overseas with the money, and all those people paid you and didn't get any thinner. And they're there to be the suckers for the next person that comes up with a scam. So regulation, I'm not too worried about that. If you want to go ahead and take a few minutes here, then we're going to start on photosynthesis. So with photosynthesis, on this one, photosynthesis is when you use light to make chemical energy that's stored in sugars and other organic molecules. So with cellular respiration, you take sugar and oxygen and break it down to carbon dioxide and water. Photosynthesis is going to take carbon dioxide and water and glue it together to make sugars and oxygen. So it's the opposite set of reactions. So something is considered an autotroph if it's able to feed itself. It basically produces organic molecules from the carbon dioxide and other things in the environment, not from other living beings in there. So that's why they're producers, because it's not made from other living beings. Heterotrophs are going to obtain their organic molecules by consuming compounds produced by other organisms. They're consumers. So you and I are consumers. We can't produce food out of light and nothing. Photoautotrophs use the light energy to derive the synthesis of the organic molecules in there. So not all autotrophs are photoautotrophs, but the majority of them are. So the chloroplast is where you're going to have photosynthesis occur in the plants. And you're going to have some pictures similar to this. That's part of the reason that I put the labs to do online, is the labs should help you in going through this and studying photosynthesis, because they're basically the same material in the worksheets help you study for it in there. So inside your chloroplast, you've got chlorophyll in there, and it's a pigment that's on the inside. The mesophyll is going to have these microscopic pores where you're going to be able to have carbon dioxide go in the leaf and oxygen exit in there. The roots, their job is to pick up water and minerals out of the soil, and it's going to deliver it to the leaf through the veins. But the photosynthesis occurs in the leaf in the green parts. If the part of the plant is brown, it doesn't do photosynthesis. It's just the green parts that do that. So the stomata are going to be these little holes in here that are going to have fluid in there with from the cells in there, and that's going to be where the holes are. The stomata are the openings to the holes where this goes in and out. So inside you've got these membranous sacs in here, and the thylakoids are the system of membranous sacs in there, and they form, it's going to segregate the stroma, which is the fluid in between, and then they form these grana or granum, which are stacks of sacs. So basically what happens with photosynthesis is you take in the carbon dioxide in the water, you rearrange the atoms in there to make glucose or sugar, water, and oxygen in there. So what you actually produce is a 3-carbon sugar in there, and then you have enough of these 3-carbon sugars that get put together to make glucose. But to simplify the equation, they look at the very end product here and just make glucose out of it in there. So this picture is your good overall summary picture of photosynthesis. So first thing that's going to have to happen is you're going to have these light reactions that is going to take the energy from light in there and make ATP and NADPH and some oxygen. NADPH is similar to the NADH that you use in animal cells. This is like a reverse electron transport chain here. Then you notice this Calvin cycle is a circular thing. Yes. So where in the Krebs cycle you're pulling off carbon dioxides, in the Calvin cycle, you're gluing on carbon dioxides in here that you're going to have sugars leave. These 3 carbon sugars that are going to leave and finish getting glued together, just like with glycolysis, where you're breaking down to make 3 carbon sugars. It's kind of like going in the opposite direction. So this whole cycle is almost like the reverse of what animal cells do. So oxygen is given off by the plants, and it comes from the chloroplasts when they split water into hydrogen and oxygen in there. So plants that don't get water very often are not going to be able to do as much of this in there. They need to have the water. And that's why they die in my house, is they don't ask for the water in there, and they somehow expect to still get it. Even those have died. They don't go years without water. So this same pattern of using carbon dioxide and water or something similar where it replaces the oxygen with some other molecule. This is the general equation for it. Plants do this where they, instead of having X mystery substance here, they have water. Sulfur bacteria use sulfur there for their electron acceptor. And there's all kinds of other bacteria that put other things in that place of it there. So photosynthesis is going to be a redox reaction in there because you're going to be moving electrons in there. So this here basically is stating that we've got the 2 phases in there. Your light reactions are the ones where you're going to convert the solar energy into NADPH and some ATP in there. Photophosphorylation is how you make that ATP in there using chemiosmosis. So it's just like what you did with the electron transport chain. Then the Calvin cycle in there is going to be where you glue those carbon dioxides back on. Carbon fixation is that initial incorporation of the carbon into the cycle. So we just call the first step carbon fixation, where you're putting it into that cycle in there. So when we look at how we get this energy from sunlight, when we have all these different spectrums, the electromagnetic spectrum here, you've got different wavelengths in there. The wavelength is the distance between the crests of the waves in there. So you've got from gamma rays down at one end, radio waves at the other end. Visible light is just a small sliver of it in here. So there's all these waves out here. We can only see a small portion of what is actually in our world. So shorter wavelengths to longer wavelengths go this direction. Higher energies with the shorter wavelengths, lower energy with the longer wavelengths. The way I remember it is you've got little dogs that are all high strung in there, high energy, barky, yappy. Bigger dogs, a lot more mellow in there. That's the way I remember the relationship. So your visible light goes from about 380 to 750 nanometers in there. Light is kind of a strange thing. It will behave as a wave, but also as a particle in there. So the photons are the discrete particles of light. They're not actually something that's tangible, which makes it even more fun to get that idea into your head in there, but they do have a fixed quantity of energy, and it's going to be inversely related to the length of their wavelength. So your photosynthetic pigments are going to absorb light. So different pigments will absorb different wavelengths of light, and that's why things appear different colors to us, is what you see is what's not being absorbed. A way that you can measure the ability of a pigment to absorb wavelengths is with a spectrophotometer in there, and then you can plot it out to make an absorption spectrum in there. So this is just showing that happening in there. And what you end up with in chloroplasts is they've got 3 pigments in there. Chlorophyll A, which is going to participate directly in your light reactions. Chlorophyll B is an accessory pigment. and the carotenoids are a group of accessory pigments in there. And your action spectrum is going to be the relative effectiveness of a wavelength in driving the process in there. So different pigments are going to need different wavelengths to drive the process. If you have a pigment that needs light that's around 600 and you give it light that's about 700, it's not going to drive the process. It's not the right wavelength for it in there. So when we look at the colors, chlorophyll A is more of a blue-green, chlorophyll B is more of an olive-green. And this is showing their absorption spectrums on here. And then the carotenoids are shades of yellow and orange. They help to broaden the spectrum of photosynthesis. So if you have a wider range of colors in the leaf, it's able to absorb a wider range of energies in there for doing photosynthesis in there. When plants turn yellow, the leaves turn kind of yellow and orange in there. What's happening is these pigments have dried up and gone away, and now you're seeing these pigments be left in there. They were always there to begin with, it's just the other ones are out of the way so you can see them now. So another thing that having the variety of pigments in there like some of the carotenoids can do is give photoprotection in there. Basically you protect the plant from getting a sunburn in there. And so one of the things in my prior quest to see if I could get plants to live in my house was I thought, well, maybe it just needs to be by the window more. Nope, you can burn a plant. I found that out. Yes, so that, well, I tried that. After I burned it, I thought, oh, it's a little dry. I'll give it some more water. It's like, are you just... Yeah. The plants realize when they come to live with me, they've gone to hell.
I used to be really good at doing stars on
So basically what happens is the light energy comes into the chlorophyll in there, and you put all this energy in so the electrons get excited, one of them spins around, and it leaves. Goes up to this excited state. And it's like, oh my God, what am I doing here? I have to come back down. So it will do that, and it can either create some heat, release some energy that can be used for ATP in there, or they can do fluorescence in there with it. But eventually it's going to come back down in there. And that's basically what the sunlight is doing to the chlorophyll in there. So the afterglow or fluorescence is when it emits light as the electron drops back down. So you can see that with some plants. I have never actually seen one that does that. I don't think we have many of them here. I would suspect they need to live somewhere that they actually get more sunlight than here if they can do that. Yeah. So a photosystem is going to be the protein complex that's going to have the light harvesting complexes in it. In there, your light harvesting complexes are going to be the pigments bound to proteins in there. And the primary electron acceptor is going to be the molecule that's able to accept the electrons and become reduced in the reaction center. So this is showing the photon light come in here. Like the electron transport chain, energy getting passed along in here. It's going to come into this reaction center complex. You're going to have the primary electron acceptor where this electron is going to go up to. So when it gets excited and it leaves, this is kind of its little plank that it's going to stand on when it leaves in there. So there's a couple different ways that this can happen. You can have photosystem 2 and photosystem 1 that are going to absorb different wavelengths of light in there. And different plants will use different photosystems. So another reason why those tropical plants don't really like to live in my house. We don't have the right wavelengths of light for them. So photosystem 2 functions first. They were named in the order they were discovered, not in the order that you use them. So if you're going to use the system that has linear electron flow, you use two photosystems. So it's going to go through and it gets excited through photosystem 2. And this one is going to absorb wavelengths at about 680 nanometers. Goes up to the primary electron acceptor. So your electron's kind of sitting on the plank and it needs to come back down. Instead of jumping back down to there, it decides it's going to kind of get on this slide of an electron transport chain that's going to pass it over here. It's going to release a little bit of energy as ATP on the way. And then it's going to get excited again and jump up to another plank and go down another slide here. And it's going to produce the NADPH there. So it's linear electron flow because it starts at one end, ends up at the other end. So the other option in there, here it's giving a detailed explanation of way more than you need in there if you need to drill that into your head with other things. The other option is cyclic electron flow. This one you only have photosystem 1. So you're going to excite the electrons, they come up, and then they come back down to the same place to do the same thing over again. So they come back to the original place. So linear ends up at a different place, cyclic electrons end up at the same place in there, and you just have one photosystem in there. So this here gives a little bit of a comparison between the mitochondria and the chloroplasts. And there, this is showing the picture in here, your electron transport chain in there, and your comparisons. So with the Calvin cycle, yeah. So here you have your carbon dioxides that are going to come in. You're going to put them in one at a time. It's showing 3 of them here, but they enter one at a time. You've got this Rubisco here, your leftover from before, that it's going to add on to and make this intermediate in here. They rearrange again, just like it did with the Krebs cycle or citric acid cycle. And you've got these phosphates on here. You've got 6 of these. They each have a phosphate. You're going to pull them off, or you're going to add in the phosphates in here. You're going to add more of them in, pull it off of the ATP. Now you have something that's got bis phosphoglycerate on here. So it went from having 1 to 2. You're going to move some electrons in here. So 6 NADPH in here. And you come down to make this glyceraldehyde 3-phosphate. So some of these glyceraldehyde 3-phosphates are going to leave down at the bottom here. When they leave here, this is where they go and they get glued together to make sugars, basically to do the reverse of glycolysis. So here it's fixing them up, putting them on these other intermediates. You move them down the cycle. And now you've got these 3 carbon sugars that get to be glued together to make 6 carbon sugars. What's left over has to be replenished so that you can start the cycle over again. From there. So again with this one, I would not bother to learn the intermediates in there, but basically that you've got 3 phases in there. First one, your carbon fixation, where you put the carbon dioxide onto the other products. The reduction, where you make these 3-phosphoglycerates that you're going to pull off to be glued together to make sugars. And then your third phase, where you're going to regenerate to make your carbon dioxide and RUBP to begin the cycle all over again. You will run into, I'm not going into photosynthesis in as much detail because you're not going to run into that as much. The cellular respiration you're going to see over and over again. So you want to get the big picture for the photosynthesis, don't worry about the intermediates. So sometimes plants live in situations that are not good for carbon fixation. So arid climates in here. So in arid climates, your stomata are going to lose water as well as allowing the carbon dioxide in. Water slips out the same hole in there. So what these will do is they're going to close their stomata on dry, hot days. It's going to decrease their photosynthetic yield in there, and you have carbon dioxide decreased in the air spaces. So what they end up doing is trying to do this wasteful process of photorespiration in there, trying to use oxygen in place of it. And it's not going to be very good for it. So the plants have adapted other ways to deal with this situation. So this is plants in general over years that have done this. A single plant cannot adapt when it comes to live with me that was not already made to live in an arid climate with water. So the single plant living with me, it doesn't get to learn that. I would need to be around for a long time with plants to change them in there. So the C3 plants in here with photorespiration, your first organic compound in there is going to be from the carbon fixation as a 3-carbon compound. These end up producing less sugar because if they're not taking in carbon dioxide, they can't do carbon fixation to make more sugar in there. So they just produce less sugar as a result of it in there. And it splits up and it starts producing these 2-carbon compounds in there and consumes oxygen and produces carbon dioxide in there. So it starts to consume ATP instead of generating it. So the C4 plants in here, we've got a couple different ways that they will adapt, and the C4 plant is one of the things in there. It gives it an alternate mode in there. What C4 plants do is instead of having everything occur in one place in the plant, it compartmentalizes things in there. So you have one part of the plant that's going to do the carbon fixation in there, another part of the plant that's going to do the rest of the Calvin cycle in there. So it's separated between these two areas. You've got the bundle sheath cells that are going to be around the veins, and then the mesophyll cells between the bundle sheath and the leaf surface in there. So basically by separating things, it allows it to be able to survive in a more arid environment in there. Again, I wouldn't worry about trying to beat up the details in that one. Another thing that it can do is the CAM plants in here. And it stands for crassulation acid metabolism in there. What these are going to do is they're going to open their stomata at night, close it during the day, and that way it will do one process in the dark, another process in the light. And it does that as a way to protect it. So pineapples are an example, as well as a lot of the succulents and cactuses will do this. So this is showing where you've got them separated here. This is just as well, we're going to do them at different times of the day so that we don't have the competition. So any questions on that part? So photosynthesis, the importance of it in there. Well, obviously if there's nobody to make oxygen, we're not going to be around very long. So the only part of the cell that's doing photosynthesis, or the autotrophic part, is the green part. The rest has to get its sugars imported from the leaves. Some are going to form sucrose for other anabolic pathways, so you'll use your intermediates to make other things in plants, just like we do in animal cells. In animal cells, you'll tend to store some of the sugars as glycogen in there, and the glycogen stores in your muscles for a source of energy. Plants are going to form and link it into cellulose that you can't break down. So it just makes a different product in there. It will also stockpile some of the extra sugars as starch. So that is photosynthesis on there. So that section's a little bit shorter.